Good evening. My name is Josh Brumberg. I'm the interim president here at the Graduate Center, and it is my pleasure to welcome you to the final City of Science event in this fall's schedule. The Graduate Center, as our name implies, is a national leader in graduate education, including the sciences, at the master's and especially the doctoral levels. As a public university, we, along with our associated Advanced Science Research Center, are committed to the idea that our scientific research has a role in advancing the public good. I am confident in saying that no other graduate school in the country takes more seriously its public responsibilities or its mission to advance knowledge for the public good. The Graduate Center is not just a place dedicated to advanced education and research, it is also a laboratory of ideas which delivers the finest research and scholarship to and far beyond the 5 boroughs. Each year, our doctoral students teach more than 125,000 undergraduates at the City University of New York, bringing the very best research and learning from the seminar room into the classroom. We are also an incubator of scientific ideas reflected in our academic programs in our free public events such as what you will witness tonight. I hope that you will make a habit of attending our public programs, lectures, and events which will allow you truly to be part of the City of Science. Tonight we are thrilled to be joined by Nobel laureate Dr. James Allison and Dr. Padmanee Sharma, who is the Director of Scientific Programs for the James Allison Institute at the University of Texas MD Anderson Cancer Center, who both will be in conversation with noted journalist Lesley Stahl, so we can all learn about breakthrough medicine and cancer research. We are bringing this conversation to you in partnership with the Cold Spring Harbor Laboratory and its BGI Nobel Laureate Archives program. This is our second annual event with them, bringing a Nobel laureate for a public conversation on our stage. It is my great pleasure to introduce Dr. Bruce Stillman, President and CEO of Cold Spring Harbor Laboratory, who will say a few words to introduce our distinguished guests. Thanks, Bruce.
Thank you, Josh. So welcome, and it's really a great pleasure to once again do a partnership with CUNY. We have an interesting partnership with CUNY, not only these events, but also high school education at the City Tech campus in Brooklyn. At Cold Spring Harbor Laboratory, we have a program funded by BGI, as you heard, that collects the archives of Nobel laureates, but equally important, promotes the science to the public of Nobel Prize winning science. And this is the second in such events that we have, and we're very, very pleased to have Jim Allison with us this evening. He won the Nobel Prize for research that you're going to hear about tonight, the Nobel Prize in Medicine, in developing a completely new and entirely unexpected therapy for cancer that has cured, as you'll hear, many, many thousands of patients in lots of different cancers. In fact, he is so prominent, he's at the MD Anderson Cancer Center in Houston, Texas, and they've created the James Allison Institute there that is going to focus on immunology and cancer and other diseases. And so Jim, I first knew when he was at Berkeley. He then moved to Sloan Kettering, Memorial Sloan Kettering Institute in New York, and then back to his native Texas at MD Anderson, where he's leading this program, along with Pam Sharma, who is trained in immunology. She's an MD, PhD, trained in immunology, and then did medical oncology, and has been very involved with the clinical trials on the technology and the ideas that Jim Allison started. And we're very pleased to have Lesley Stahl moderate this evening. Lesley, as you know, has had a long 30-year career with 60 Minutes. She's one of America's best journalists, I think, and has won numerous awards, including 13 Emmys. I have to put my glasses on for this. 13 Emmys, a Lifetime Achievement Award, the Edward R. Murrow Award, which is very prestigious, Alfred DuPont Columbia University Silver Baton, and the Founders Award for Journalistic Excellence. Those are just some of those. She has also written a best-selling book about reporting live, and Lesley is a great supporter of Cold Spring Harbor Laboratory, and I want to thank you very much for doing this. So it's a great pleasure to introduce Lesley, Jim, and Pam.
I'm delighted to be here. One of the great, I hope, gifts to all of you is I don't know anything about science. So I'm going to be starting as if I'm in first grade. I hope you appreciate that. First of all, I'm just thrilled, and I consider this a real treat to be interviewing both of you. I'm just going to jump in because I heard a word from Dr. Stillman that picked my ears up. He said, cured. He used the C word. And most doctors don't like to say cured when it comes to cancer. So cured? Explain cured, Dr. Allison.
Well, people have been hesitant to use the word cured because, there have been incidental successes in people, but no large trials. But in the first trial of the drug ipilimumab that I had invented, there were some immediate responses of people that with late-stage melanoma, where the tumors disappeared, but
And never came back?
well, there was a study of over 4,000 patients that had been treated, and 20% of them were still alive 10 years after their treatment stopped. And before that drug became available, the median survival was 7 months, and fewer than 3 people were alive at 5 years. So, and since then, the good news is we can do better. Since then, another drug was developed against a different target, and the two work very well together because they're kind of different. There's a study that's about 9 years out now showing in the same disease, late-stage melanoma, with a survival over 50% at 9 years, again, compared to less than 3% at 5 years from before. So I think those patients can be considered cured, yeah. And it's happening again and again in several other kinds of cancer as well.
Dr. Sharma.
Yeah. So as an oncologist who treats patients in clinic, I think it's true that when you see a patient with stage 4 cancer where the cancer has gone throughout their body, right? It's not just in one place, it's in multiple places. We're reluctant a bit to say the word cure when we give treatment because we know that the cancer can come back at any time in any one of these areas. I think with what Jim developed in immunotherapy, with immune checkpoint therapy that you'll hear more about, is that you give a drug through an IV. That drug circulates in the body, and then it allows your immune system, the drug does not target the cancer itself, it allows your immune system to then get activated and target the tumors. And because your immune system circulates everywhere, it can go everywhere that there are tumor cells. And then those immune cells eradicate the tumor cells because that's what your immune system does. It gets rid of things that do not belong in your body. And so we found a way to then take advantage of your own immune system to treat the cancer. And the other thing is that your immune system can have memory responses. So when you get a vaccine, for example, for polio or smallpox or those things, you have a memory response so you don't get that disease. So with these cancers, we're hoping that you have a memory response, which the majority of patients tend to do, so that the cancers can't come back. And that's why we feel comfortable using the word cure in some of these situations that you'll hear about.
Is immune therapy like a vaccine? Is it similar to a vaccine?
Both of them involve activating the immune system. Vaccines typically are considered to prevent something coming on. So you get a piece of whatever, a virus or something, and you immunize against that, and you get an immune system in advance so that it can actually protect you. So this uses the same sorts of cells that are there, except there's two kinds, B cells and T cells. B cells secrete antibodies that are going around the body, and typically that's what protective vaccines generate. What we're doing is unleashing these other kinds of cells called T cells that actually go out and directly search through your body. T cells are very cool. I first learned about them as an undergraduate when they'd just been discovered and nothing was known about them. And I asked the professor, he said, well, there was these new cells that were thought to play a role in immunity. And I said, well, how do they work? How do they see what they're going to see? And how, they percolate through your tissues, through your blood, and just scan your whole body. And how does that work? And he said, we don't have a clue. And he said, and I said, perfect, I'm going to work on those. And so that was in '68 about, and I've been working on them ever since. And we've still got a lot to learn. But There are other kinds of immunotherapy now that also try to use T cells. What we do is unleash T cells in the body. What I've done is study step by step the molecules that are involved in regulating them. And I found this molecule is the brake and reason that it might stop an immune response to protect you from getting harmed by it before it eliminated the cancer. So I found a way to suspend the brakes temporarily.
Is cancer a disease of a defective immune system?
No, not necessarily. Cancer cells have antigens on their surface that can be detected, but normally it's a complicated process, but there are a number of other signals that an immune cell has to get activated other than just the antigen. And tumor cells don't have those, and so we have to alert them that there is something there that the tumor, all tumor cells have mutations. That's what ultimately causes cancer, and those mutations cause change in structures.
Not the immune breakdown.
Yeah, and so the immune cells are after that, but they don't have all the molecules that are necessary, so they're just regulated by normal processes.
If you think about it, your own body has mechanisms that stop your immune response. So if you get the flu virus, you'll have an immune response to the flu virus, and then the immune response stops, and you get rid of the virus for that period of time. Those mechanisms that stops the immune response are the things that cancer has co-opted. So it's used the same mechanisms so that the immune system is no longer seeing the cancer. And so you have to retrain the immune system with these drugs to get them to recognize, hey, those cancer cells are trying to fool me, but they really should be attacked. And they're using the system that your body normally uses so as to not to attack your normal self, right? You don't want to attack your heart or your liver or your lungs. Those are normal. But the cancer is using those mechanisms to protect itself so that the immune system doesn't see it. But when we train the immune system, we can change that.
So take one step back. Tell us when you say that it's a system, an immune system, what is that? Is it hormones? Is it fluids? Is it
Part of it. It's a series of cells that your body has developed to protect you, mostly against viruses, bacteria, things like that. Also cancer, if we help it a little bit. But it is cells that cruise through your body. They communicate with each other and with your normal cells by cell surface contact and things called cytokines, which are like hormones. And your normal cells can make hormone-like things that influence the immune cells too. And it's all held in balance by mechanisms that we're still trying to understand. They keep it, allow it to protect you against viruses, to protect you against bacteria, and when activated properly can eliminate cancer. In some cases prevent it perhaps. The HPV vaccine, for example, is one that does that. And do that without killing you. And that's the cool thing about it, is
that's
that
probably important.
it can kill you, but it has all these protective mechanisms in so that it doesn't.
Well, if you have cancer up until now, you usually got chemotherapy. And the chemotherapy would make you so sick that, it was almost worse than getting cancer. Some people could feel that way. When you give your immunotherapy, what happens to people? First of all, is it a drip? Is it an infusion? And do people get sick?
It's an antibody, and it just goes in. You sit for. I actually was interviewed by a journalist one time who had melanoma as she was getting the drug, and it's an infusion. Yeah, and you sit there for about an hour and a half, and it drips into the arm, and then that's it. You go home. There's nothing.
One shot?
Most of the time it's 3 or more, but it can be 1, I think. But we won't.
Did
we
they
won't
go
there.
home and just lie there moaning and groaning?
No, usually, some people that, immediately or sooner or later felt better. In the early days, there was a patient that a colleague of mine at Sloan Kettering had that was told before he got it that he was hospice bound, and so he got this last thing, and the guy said, okay, I'll take that, and he took it, said, I'm going to go on a last motorcycle ride, and he called his oncologist a week later and said, I'm in Denver and I feel great, and turned out his tumors were completely gone when he came back. But very often there are inflammatory and autoimmune things. It can be quite serious, but clinicians who have learned how to deal with them Most cases, they're very, they're not the overwhelming sickness that you get from chemotherapy. And with the chemotherapy, basically, it kills tumor cells a little bit lower doses than it kills normal cells. And it's given a lot because unless you kill every last tumor cell, it's going to come back. What we know now is that you don't have to kill all the tumor cells initially. You just have to kill enough, say with a tumor cell, it becomes like a vaccine then. That's enough to alarm the immune system that something's going on, and the T cells will take it out after that and then give you the memory that is needed. So,
Well, you see patients, Dr. Sharma. Tell us what you see when you give this treatment.
So the immunotherapy drugs, they're very different from chemotherapy drugs. When we give chemotherapy drugs, for example, we expect 100% of the patients will have a side effect because it's chemotherapy and it's going to cause side effects. When we give the immunotherapy drug, we expect about 10% or so of patients to have side effects. The majority of the patients do not have any side effects from when we give a single immunotherapy drug. And we give it through an IV infusion, as Jim mentioned. You sit in the clinic and then you go home. You're not admitted to the hospital to get the drug. When you do get a side effect, though, just remember what before, when we're telling the immune system to act against the cancer, that's what the immunotherapy drug is doing, that immune system can also act against your normal cells, because now we've taken the brakes off, right? We've removed those normal brakes. And because it can do that in some patients, it will cause an inflammatory reaction in certain organs. So for example, it can cause inflammation in the skin, so you get a rash. It can cause inflammation in your colon, so you can develop diarrhea. And these things can be managed. We definitely know how to manage them now, and are very good at making sure we stop the drug, and we give steroids as needed to control the inflammation. When we give two drugs combined, you see higher frequencies, or more patients will get the side effects and toxicities. But we also know that when we give two drugs combined, we also have better efficacy against the tumors. So sometimes it's weighing these things to decide when to give one drug or when to give two drugs. And that's a discussion with your oncologist, depending on how you're doing.
So I heard, tell me if this is true, that when you first started trying these in the clinical setting, regular cancer doctors, when they give chemo, they expect to see the tumor shrink almost immediately, right after the treatment. But what happened with your situation? They didn't, right? Tell that story.
well, what we knew from the mouse studies is we're initiating a response. You basically, the way your immune T cells work is you've probably got 100 million or more different ones. And a few of those might be able to react with the tumor cells. So you've got to expand those to get an army, that expand, of hundreds of thousands or a million copies, of that same clone. It's got to have the army to go. And that takes some time. And so there's a lag time before it could happen, before it could attack the tumors.
So you'd give your treatment and it wouldn't happen quickly?
Yeah, it could take a while to come along.
Okay.
In some patients, it might take a month or more in the early studies when it's given by itself. Whereas chemo, the chemo drugs are poisons, and so if the cancer grows at all when the drug's on board, that means that it's not working, by definition.
Didn't
and so
the doctors think it wasn't working?
Exactly, they were.
Well, tell about that.
Well, the tumor would keep going, or sometimes when people started looking, after they first said the tumors are growing, it's not working.
They kept growing?
Yeah, they would get bigger and bigger, and they said, oh, it's growing, we got to, they failed, we got to go to something else. In some cases, when they actually stuck a needle in it and looked at what was in there, it was just immune cells. There were no living cancer cells left. And so there were two things going on that fooled the doctors. And so that was called pseudo-progression. It was actually the immune system killing the tumor cells.
But the tumor kept growing.
But there was no tumor. What was called a tumor was growing, but it really wasn't tumor even. So those two things confounded some of the early work. So they'd used.
Patients stopped trying it because they thought it wasn't working?
Some did, I think, but people kept on, and there were studies that patients would go back to their home communities, and they'd come to New York, for example, from wherever, and be treated, and doctors said, it didn't work. Your tumor's still growing. They go home, get chemo, and then the word came back, oh, the drug doesn't work, but it's sensitizing them against the chemo. And I said, no, it's just working, And finally, it was.
When they got home, they realized.
Yeah, yeah. And then they would go away, but at first they thought, oh, it's because what their doctor gave them, which are drugs that didn't work by themselves. So.
Wow.
Anyway.
Yeah, because usually in the clinic, you're going to have CAT scans about every 3 months when you're getting treatment. And with chemotherapy, you'll see that at the next CAT scan, within that 2 to 3 month period that you're getting it, chemotherapy will work immediately, and you'll see that result if the chemotherapy is working.
Yeah.
When patients in immunotherapy, when we would do the CAT scan about every 2 to 3 months, sometimes you wouldn't see the tumor shrinking, or sometimes you could see it growing. But if you continue to watch the patient over time, you will eventually see that in later CAT scans, that tumor is definitely shrinking and patients are doing better. One key that we look at in the clinic is not so much just a CAT scan. We're not treating CAT scans, we're treating patients. So if the patients tell us they're feeling really great and they're doing better and they're starting to enjoy their life more, sometimes you look at the CAT scan, you say, maybe it's not really growing. Maybe what's happening is all those T cells, all those immune cells are infiltrating into the tumor to get rid of the tumor, and so let's just wait to see how the patient is doing.
So let's step back. You start your research, and as I understand it, and this happens with a lot of breakthrough science, there was so much skepticism about what you were aiming for, and naysayers. And despite that, you kept going. So talk about the way the general community that you live in thought you weren't going up the right tree and you didn't stop.
Well, I think what was going on is that as molecular biology came along, people began to realize, let's study how normal cell growth is regulated and all that, and if we could figure out what's different about cancer, we could treat it by hitting the cause, the mutations that are causing, and so that made a lot of sense to do it that way.
To you?
To me too. I mean, I thought
only you.
But to me, I mean, I knew what the immune system was capable of, or thought I did, and I just thought if we knew what controlled it, and we could unleash it when we need to for cancer, to attack cancer cells, then it would figure out what to attack. It would take care of it, and so we started working in mice as we worked in tissue culture, then worked out in mouse models what the different controlling things were. Sometimes I did many things, some of which worked a little bit, slow a tumor down a little bit in mice. We have experimental tumors in mice that are cell lines that grow forever and you could take a whole lot of genetically identical mice and give them this same tumor and then compare it. And we'd get some things that would slow them a little bit. But when we first tried this CTLA-4, that's the name of the target, the antibody to that, the tumors just went away when we injected it. It took a while. Yeah, in the mice.
Wow.
And it happened with every tumor model we did. It didn't really matter what the kind of mouse was or what kind of tumor it was, whether it was a lung cancer or colon cancer or leukemia.
Wow.
We were able to see all of those. And at that point I said, we may be onto something here,
But this idea of using the immune system has been around in the field of cancer research at least 50 years.
The first person to propose it was Paul Ehrlich, who was a biochemist in 1906, actually.
Took a while.
Yeah.
Yeah, but why? One of the very first stories I ever did for CBS News in 1972 was this. It was going to be the cure. They were going to do immunotherapy in 1972. Nothing happened. So what was different about the approaches that other researchers were doing at that point that changed?
I think that a lot of the other approaches were based on good ideas, but we didn't have sufficient knowledge of mechanisms at the time. And so rather than just start throwing things at the cancer, what I decided to do was understand how the growth of a T cell was regulated, to find the molecules that did it, the pathways, as best as we could tell. In other words, the buttons to push on.
We have to step back. I don't know. Does everybody know what a T cell is? I know you talked about it before, but I don't know what a T cell is. So
It's
can
a
we-
white blood cell that protects you. There are actually several kinds, but anyway, they protect you against. If you don't have them, as in HIV/AIDS, you're in bad trouble. You get cancers, you get infectious diseases, things like that. They're the things that prevent that, and if you lose them, you're in bad shape.
And people have lost them.
Yeah, in AIDS, that's what it is. You lose those, you don't have them, because the HIV virus kills a particular kind of T cell, and then they're.
That's what you did. You studied the T cells
Mm-hmm.
in the beginning and learned the mechanism.
Yeah.
And there are switches that you figured
Well,
out.
there's one. It's pretty easy to explain. There's one called the antigen receptor, and that's the thing that it uses to know what it ought to react with. It's kind of like the ignition switch in a car. Like I said, you've got maybe 10, maybe 100 million, we're not sure, different ones in your body. Each one, or small numbers, clones have a different switch, if you will. And so that we worked that out in the early 80s, that molecule, and then found out that there wasn't quite so simple. There was another one. It's kind of like the gas pedal, a molecule called CD28. And we worked that out. Anyway, the next one we quickly found was CD4, and it's the brakes.
Oh.
And we studied earlier trying to do each one, but they didn't work so well. But when we found the brake, we found if we took the brakes off, when there's a tumor around, the immune system would see that as foreign and kill it. And that's all we had to do is take the brakes off.
Oh, my goodness.
And
Finally.
you have T cells that can recognize virtually anything, but they're not because they're held in check, or they get started and they stop because if it's a cell thing. But anyway, that was basically the idea.
Was there a moment in your lab, aha, the
Yeah.
great bathtub story?
Well,
Except you were in the lab?
There were some aha moments when we finally figured out what those molecules did, but there was a big aha when I looked at the mice that we'd inject with the antibody, and it was actually an experiment where we had a lot of mice, and half of it had been treated with it. They all had tumors. We gave half of them the antibody and half something else, and I went in actually to look at the mice, and I saw the tumor shrink and go away in half the mice. I didn't know which ones were treated, but it was pretty clear to me that half of them, that was probably the experimental group and not the other one. And so that was an aha moment.
So you're in the lab, you're working with mice, you eventually get to people. You had to get FDA approval first.
Well, the path to people, though, was interesting because we did the experiment I'm telling you about in 1994. We published it in 1995. It wasn't until 2011 that it was approved.
Whoa.
By the Food and Drug Administration. It wasn't until about 1998 or 99 when we could finally find a company that was willing to help us by helping us make the drug that we needed to repeat in humans what we've been doing in mice. We couldn't use the same drug in humans. So we had to make a new drug.
Why couldn't you use the same drug?
Well, it was an antibody that we'd made, and it's a mouse protein.
Oh.
If you inject a mouse protein in a human, it's foreign, and it'll cause problems.
But
So
it could ignite an immune response.
it would. It would. If we injected mouse protein in humans, it would to itself, and then you might get kidney disease because your protein
Might kill
would-
you, but you'd have an immune response.
So what we found was a company actually that had a mouse that replaced the mouse genes with human ones. So those mice actually made human antibodies, and we could use those then to make the drug. And so that's what we did.
Does
The
it
first
still-
trials were about 2001 or so.
Does the medicine still come from mouse altered?
A lot of them still do. Some of them are made strictly by genetic material, in tissue culture, but a lot of them still come from making them in mice. They're not necessarily produced in mice anymore. They can be made in cells in tissue culture, made like you make beer or something, actually.
I don't think maybe we're learning too much. Is this true? This is, reporting. I don't know. I need to get my source to tell me it's true. That the reason you wanted to go into cancer research was because of your mother?
Yeah.
So tell us that story.
Well, when I was little, my mother was sick a lot, and I didn't really know what she had, and I was called in once and they said, your mother's very sick, and I was actually holding her hand when she passed away. And it wasn't until several years later, I just knew she'd been sick a lot. I knew she had some burns on her neck and scars found out that she'd had lymphoma. And and then shortly after that, one of her brothers got lung cancer, and shortly after that, another one of her brothers got melanoma.
Oh, boy.
And so, and then years later, when my brother got prostate cancer, and so, that's the reason. So I always thought, grew up thinking, if I could, I'd like to do something about this, because I'd seen chemo. I'd seen cancer, and I'd seen chemo, and I'd seen radiation and surgery, and thought, we ought to be able to do better than this.
We've all thought that, haven't we? We've all thought, they have to figure out something better than chemotherapy, and at last. So can you tick off the kinds of cancers that that we know already this therapy works on?
Oh, God, I'll give some, and you give some. So melanoma, late stage, early stage. Lung cancer, it's moving late stage, it's moving into early stages. Pam did the trials in bladder. It works in kidney. It works in several kinds of leukemia. It works in some kinds of bowel cancer. It works in anal cancer. It works in what?
Wow.
It's easier to name the ones that it doesn't work in at all. And
All right, let me ask it that way.
And there's two of them, and that's glioblastoma and pancreatic cancer.
Oh, pancreatic,
And
come
those
on.
are the ones that we've got to do something about. And we're slowly through the trials that Pam does and other things, figuring out, I think, getting clues at least as to why what works in other tumors doesn't work there.
So, but do you think eventually that this will work in pancreatic and all cancers?
No, well, I think it'll work on some patients with all cancers. I think we'll be able to cure cancers. The numbers are getting up there. Things I mentioned earlier in melanoma now we're up to perhaps 80%. In lung cancer, there was a recent trial, an earlier trial of a type called neoadjuvant. Pam actually did the first trial of that sort with these drugs earlier on in 2006, but a recent trial like that in lung cancer had a response rate of 77%. And so, it depends. But I think that's what our institute is all about. We know the rules. We know some of them it works better than others. We can learn from that. And the ones we're learning from the ones where it doesn't work, why it doesn't. And, the good news is we know we can do it in some cancers, so it's a matter of just working harder and getting it in more. I would not say we're going to eliminate cancer. I wouldn't. And one of the things I want to also point out is that it's not that it's going to completely replace chemotherapy or radiation. In some cancers it will, but in other cancers I think it can really be helped if you do a little bit of radiation and a little bit of chemo, but you don't have to try to kill every last tumor cell. You can just give one treatment, maybe kill some tumor and stop, and let the then do what we do and let the immune system finish it off. So you don't have to make your hair fall out and all that stuff. You just get one dose. And radiation's been the same thing. You radiate one tumor, and you can get all of them to get rejected if you give the drug,
Yeah,
in some
part
patients.
of the arsenal, as they say.
Yeah.
What's so difficult about pancreatic cancer?
Well, it doesn't have very many mutations. That's what's known as mutations. You've got in melanoma, you've got a lot of mutations because it's caused by being in the sun and ultraviolet damage. Lung, bladder, esophageal, some other cancers are caused by smoking and stuff. And
That's
smoke.
easier?
They are easier because they got a lot of targets. I mean, it's kind of a surprising thing to me that the cancers which were the trickiest to treat by more conventional means because they had so many mutations that by virtue of those mutations could escape what you threw at it. But that makes them a sitting duck for the immune system. So those are the easiest ones. Yeah.
All right, I'm going to ask you if you want to write questions down. We'll be collecting them shortly. But breast cancer? Did you mention breast cancer?
Yeah, there is an approval in breast cancer as well. And again, it's combination with chemotherapy. So as Jim was saying, sometimes you don't have to give the immunotherapy alone, or you don't have to think that immunotherapy will replace chemotherapy. Sometimes it will be given in combination in certain tumor types. The other thing, to remember is that even though we have FDA approvals in melanoma, lung cancer, breast cancer, bladder cancer, kidney cancer, only a proportion of patients are responding. So it's not 100% of any of those tumor types where we see all the responses. And so we have to figure out why that might be. And one of the things that, Jim did so much work in understanding the mechanisms of how a T cell works, right? A T cell has an on switch, it has an off switch, and you have to make sure either you're pushing in the right direction to get that T cell going. But T cells are only one component of the immune system. And so the immune system has myeloid cells, natural killer cells, B cells. All these other cells are part of your white blood cell system, which are part of your immune system. And we still have to figure out the mechanisms of how each of these are working and how we can then combine therapies to target T cells and B cells maybe in some tumors, or combine therapies to target myeloid cells and T cells in some tumors.
Yeah, in pancreatic cancer, what you asked about specifically, there are a lot of those myeloid cells that Pam mentioned, and those typically turn off T cells. They're made to function to protect wounds, and in doing that, they sort of keep the immune system from causing any further damage. So they'll actually turn T cells off. But if you treat them right, we know from some work that we've done in mice now that you can also do things, and we don't exactly know what's happening in the myeloid cells, but there's certain things that we could do. They'll help the T cells kill the tumor, so we can make friends of them. And that's one of the big challenges, I think, in pancreatic cancer and in glioblastoma, which also has a lot of those kinds of cells, is how do we turn them around where they become helpers to the immune system instead of turning it off.
So it sounds to me as though you're continuing with this research with the goal of getting at every kind of cancer?
My personal goal and the goal of the Institute, as stated, is to keep going and try to, cure as many cancers and as many patients with as many kinds of cancer as we can.
Yeah.
No matter what it takes.
So if you do have questions, I know there were cards given out, send them to the end of the rows, and somebody will pick them up, and then we'll get to them. When someone wins the Nobel, they almost always have this great story of how they heard. And at least half of them, at least half the times I've heard of it, they hang up because they think it's a hoax. Come on. And so tell us.
They don't answer, which is...
What happened when they called you?
Well, the stuff that I did was around, and so people every year for a while were saying, "Oh, you're going to get the Nobel." I said, "Stop it! I'm not going to get. No, I'm not worried." I certainly didn't wake up at 5 in the morning expecting a phone call ever. On the one hand, the phone rang one morning. We were actually here in New York. We'd come up from Houston. Pam was winning an award from the
Cancer Research Institute.
the Cancer Research Institute, which is headquartered here, and does a lot to fund cancer research, especially training of young scientists. But she was getting their top award, actually, and so.
This is a sad story.
So we came in, and the night before, some people were saying the same thing. Anyway, I got up in the morning, and the phone rang early, and hung up, and Pam said, "Well, maybe next year." I said, "Yeah, let's go back to sleep." and then about an hour later, my son called and said, "Dad, you won the Nobel Prize."
Oh.
Yeah.
He got up. He had been getting up at 5 in the morning and watching. He knew where the Nobel Foundation press conference was being called, so he was watching live. And he saw it.
He suspected.
And so, the funny thing about it is, not funny, but nice thing about it. Well, first of all, the word spread very quickly then. And so within an hour or so, our hotel room was full of people and bottles of this and that inebriating substance, and including a Hudson bourbon, as I remember.
Don't forget that.
But anyway, and then the press guy from Anderson MD Anderson got a hold of us and said, "Didn't you get the call?" I said, "No, we didn't." And he checked
Never
it.
got that.
No, what they had my office number.
Oh.
And I was here, not there. And so he called and gave them my cell phone number. So they called back. And so I took the call, and my son filmed it on his iPhone, you know.
Is your son here?
No, I think
No?
there he is.
Where are you?
There he is back there.
Sat with him last night.
Robert, I think he brought the Hudson bourbon, actually. But anyway, he got to film the call.
Yeah, it's pretty exciting.
Yeah.
Well, I guess you were completely overshadowed with your award. How sad. You know,
It all went out the window.
I
I think she was going to be interviewed by Time magazine, right? And
Yeah.
they said, can we put that off? So we're very happy about that.
No, I don't imagine.
It's okay. It's all right.
You know, we haven't done the full disclosure because they are partners in working on this kind of immunotherapy, but they're also married, and I haven't told you that. I waited to the end.
Yeah.
So also I hear that when you found out, after all your work, that you got the FDA approval to go forward with clinical trials on humans, that you were at a very interesting place when you heard that.
Yeah, I was at a conference on melanoma at the at Cold Spring Harbor. A group of about 30 people that were there to discuss small molecules and the molecules that attack the cause of cancer. Anyway, the precision medicine type. And I was there as sort of the token immunologist. I think there were two immunologists maybe in that there, and I'd heard that decision was going to be made, and I kept looking at my iPad, I expected my friends that had made the antibody, would hear it. Finally, I got an email with these guys. They were in California. It was 11 in the morning, and they had glasses of whiskey that they were holding, and I thought, I guess that's good news, and anyway, then it came out, and actually we had at lunch, it broke out at Cold Spring Harbor, broke out the champagne to celebrate the approval for the FDA.
Were you there, Bruce? This is very exciting. This is a question, I'm going to ask a question that I hate being asked, but I'm going to ask it anyway, because if there's a good answer, you'll all be interested. You've worked very hard. You've been in the lab constantly. Do you have any interests outside of this research that?
Well, a few. I play blues harmonica a little bit. So I play in two bands.
He's in two bands. I wanted you to hear that.
Well, they're bands largely of scientists and clinicians.
Oh, okay, forget that.
Not bad though. We still play. The one's called the Checkpoints, and the other one the Checkmates. I also though play with Willie Nelson whenever he's in the area.
He played with Willie Nelson. I'm not kidding. So you want to tell him your instrument?
The harmonica. It's very,
He
very
plays
arcane.
a harmonica. Some of us heard him play last night, and it's kind of jazzy. It's very exciting. A lot of scientists are good musicians. It's true. So I've been waiting my whole life, and I'm not a young chicken, and you all know that, for the cure. For the cure. I know you have too. We're right there, aren't we?
Yes.
Some people we are, yeah.
We're right there. What's the difference between what you're doing and what they do with CRISPR, which is, gene editing?
Yeah.
Is it like a parallel track? Or?
It's a tool that could be used to improve T cells, for example. Some people grow, instead of letting them expand in the body by playing with the brakes and stuff, other people take them out of the patients and expand them, and in order to make them not respond to those brakes, you could use CRISPR, for example, to eliminate, just take those out of the genome so that the normal suppressor mechanisms aren't there, so they'll stay alive and keep killing tumor cells longer. that sort of thing. So it's very useful.
Is it an outgrowth of what you learned about the structure?
I think the targets are, yeah. Yeah, and it could also be used, there are ways of using it to knock out individual genes and seeing what, as a purely research tool, to see what they do, you know,
But
sort of thing.
your basic fundamental research on how the T cells work, how to turn them on, how to turn them off, launched these other avenues for
Well, I wouldn't say exactly
attacking
it launched
cancer.
it, but it launched
Yeah.
a lot about the ways they might be used in cancer
Yeah,
specifically.
I think what Jim's work was definitely the way to think about cancer treatments. CRISPR is a technology that you can use for lots of different diseases. You know, they're thinking of how to use it outside of cancer. They can use it in diabetes. They're thinking of how to do lots of things because it's a technology to treat defective genes, per se.
But when it comes to cancer,
Yeah.
then they go right to the T cell
Yes.
right now. All cancers involve T cells?
I think so.
Yeah.
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 It
Okay.
used to be thought that, you know, the idea was, oh, cancer cells are so much like normal cells, you know, they're not viruses, they're not bacteria. That's not enough. You know, the immune system, they're invisible to the immune system. That's just not true. We showed that many years ago. But all you need is really, to possibly do it, is one antigen, one mutation in a protein that you can get a really good immune response to, theoretically at least. But there's never just one mutation. There are always more than that. But yeah, it's a pretty fine detection system that T cells have for knowing what's foreign.
Okay. Do we have questions from the audience? I know we have some. Here we go. What role do you see for personalized medicine to treat cancer in the near future?
Oh, that's ongoing already. I don't know if some of you may have read the New York Times headlines about how personalized medicine, whereby patients were selected, these were patients with anal cancer, rectal cancer, nail cancer, who were selected for a particular marker, and using that particular marker, that's personalized medicine. And because these patients then had what was called a MSI, or microsatellite instability in their tumors, they were selected for that and then treated with immunotherapy. And those patients had every patient basically had a response. So personalized medicine allows us to select the correct patient. And when we have the right patients, you can see great responses in all these patients because we selected based on a personalized marker for that particular tumor.
And another way to do it is based on a lot of the stuff that goes on at Cold Spring Harbor. You do the genome sequencing, and then you can detect what's mutated in the tumor, and then there's certain computer programs that can tell you which ones are likely to stimulate your immune system, and then you can make vaccines of them. It's being done now in that same virus backbone or structures that were used for the COVID vaccine. You can just plug in those sequences potentially into that vaccine and therapeutically treat a person with the exact antigens that their own tumor has and engage just those T cells. That's a lot of work, but it can be done.
But getting easier, as you and I discovered last night. If anybody has cancer, is your kind of treatment accessible? If any
doctor in the U.S. can prescribe these things. It's approved by the FDA.
I know everybody in this room either had cancer themselves or knows someone who did, who suffered with chemotherapy. And you know, the story I heard about your mother, and maybe I just heard it wrong, was that you saw her on chemo and that it was horrendous, and you said, that can't be.
I saw her brother on
Her
chemo.
brother on chemo.
He was laying in an oxygen tent, and what struck me about it was I'd known him since I was little. He was a rancher, you know, quintessential Texas cowboy, you know. He could roll a Bull Durham cigarette with one hand while he's riding a horse, you know. And that's what did him in eventually, you know, because he had lung cancer. And but I saw him go from this, you know, Marlboro-like guy, literally, to a shriveled guy in a tent with chemotherapy, you know, going in his arm, and that was not pleasant either.
No. It's so exciting. It's so uplifting. And I'll tell you something, we need these good stories right now in our lives, all of us. Can there be other approaches beside the immune system that have been considered and might be considered again? I think that's the question.
Sure. I mean, the chemotherapy is curative for some kinds of cancer, and the targeted therapies where you inhibit the mutated enzymes that cause cancer. I don't think they're going to be. Most of the time, there's too many mutations to treat, but I think with some study, they might be useful. And what's next to come along? Who knows?
Who knows? But then it sounds to me like a combination of different kinds of remedies, whatever.
Well, that's one of the things that we're trying to do is combine chemotherapy, whatever it takes. I mean, it's the bottom line.
Whatever it takes, and to make it easier. Is the same immunotherapy used for all cancers? And we discussed a little bit about that. Eventually, the silver bullet.
So I think the immunotherapy space has definitely become broader. So you have these immune checkpoint therapy agents that Jim's work really led to the discovery of, and all these new drugs in immune checkpoint therapy. And those are drugs that target specific pathways on T cells. So those are drugs like anti-CTLA-4 or anti-PD-1 or PD-L1. Then there are drugs or treatments where you take the T cells, you engineer them in a laboratory, and then you give those T cells back to the patients, and those are called CAR T cell therapies. So they're all immunotherapy, but they're different immunotherapy that work in different tumor types. So the CAR T cell therapies tend to work more in the leukemias, for example, whereas the immune checkpoint therapies tend to work more in the solid tumors.
They're designed for specific kinds of cancer, whereas I think that maybe the question is, but these other drugs that we have, the checkpoint blockers, it's the same drug across very broad kinds of cancer. You're not treating because you're not treating the cancer cells, you're treating your immune system. So the kind of cancer, theoretically shouldn't matter. I mean, it does in the real world because of some of the things we talked about, but because the different cells they've got other things going on in them that keeps the immune system from being effective. So,
what happens? Well, first of all, is there ever metastasis with your treatment, and you just give this treatment again?
Not if it works.
Not if it works. But I mean, it's not 100%.
No, and the majority of the FDA approvals so that any doctor can give the drug is in the setting of metastatic disease. So stage 4 cancers are where these drugs are approved to be used. But we think that, again, Jim mentioned that if we give the drugs earlier, and those are some of the clinical trials we first conducted, in the earlier setting, before it gets to spread all over the body, we actually see better responses when we give them earlier. So now we're looking forward to seeing some approvals also for the drugs to be given earlier.
So you don't have approval.
We do have approvals in some settings, but not all the tumor types that we have the approvals for in the metastatic setting.
Okay.
It's always at the metastatic setting first, and then it works earlier because, experimentally they figure if you've got terminal cancer, you can take more chances.
Right. But now, we were asking, somebody asked, if anybody gets cancer, can they get your treatment? Not if it's not metastatic in most.
Yeah. The first patient I ever met was a 22-year-old woman named, she was 24 when I met her, named Sharon Belvin, it's okay with her if I talk about it. But when she was 22, went in for exhaustion. A doctor said, you have melanoma. She had 30-something metastases in her lungs. Her liver was riddled with too many to count. She had one in her brain. And she didn't respond to any chemotherapy or radiation therapy to the one in her brain. And after the very first dose she was told, when she was 24, you're getting hospice, hospice, or you want to try this new drug. And so she took IPI, we call it, and her tumors just went away. They just all went away. And that was in 2006. Now she has two they're still teenagers, I guess. But,
No, I think they're a little older, but yeah.
Maybe a little older now, but anyway.
What did
Last
you,
time I saw them, they were teenagers.
so you were beginning to say the name of the drug, and it's
Ipilimumab.
That's the drug. Listen to that. Do it again.
Ipilimumab.
I can't say it. It's written down.
It's
I can't
hard.
say it.
It's hard. They all end in, because it's a monoclonal antibody, MAB, made in mice, murine. So it's MUMAB. And all of them that are made from antibodies are called MUMABs. But then whatever goes in front of that is gibberish. Ipi was, I don't know where it came from, but since it was invented while I was at Berkeley, I tried to convince them to put an H in front of it and call it hippie.
What about,
but
let's call it the Allison.
okay.
Well, when you see it on the TV commercials, though, it then has a different name, right? Then
Yeah.
it's Yervoy or
Yeah.
Opdivo or Keytruda.
Yeah.
Those
Wait,
are the
they're-
drugs
they're
you're
on
seeing in the
commercials?
Oh,
TV
yeah. There's
commercials.
commercials all the time, all the
I
time.
haven't seen these.
Yeah.
I have one final question, a final question about a particular kind of cancer, which is glioblastoma. Glioblastoma, the horrible brain cancer. Yes, no, you don't know, or?
It
It's a hard
doesn't
one.
work in glioblastoma yet. And we are trying to figure out why. And really, in glioblastoma now, there's a group called the Breakthrough Cancer Group that's doing some studies in glioblastoma. It's a team of 5 institutions working on this to try and understand glioblastoma better. And what we are seeing is that most of the other tumor types, you can go and get samples from the tumors multiple times, right? In melanoma, you get a sample, or in lung cancer, you get a sample, and then you treat the patient, you go get another sample, you treat the... In glioblastoma, because it's in the brain, we're not able to keep going back and forth so many times. And so this group came up with a new way to do getting the samples, and now we'll try to understand how the immune system is changing in that environment and maybe come up with different ways to target for that tumor to get an immune response. So those studies are still ongoing, and we have a long way yet to go.
You know, they used to say that if it was head and neck, that you couldn't take chemo because there was some barrier, and that's not the problem.
No.
There's no head and neck.
Yeah.
Well, we've finally met our hero, everybody. So thank you all. Thanks for coming.